just one point. We're fighting for space. So, good afternoon. Good evening, everyone. Our training program is on AMR, microbial resistance, and relevant to that one topic is going to be role of Phages in combating AMR. So I will try to justify my role, and then Dr. will be explaining role of Phages. So, brief introduction about antibiotics. All of you must have gone through in earlier lectures also. So, long back, the antibiotics they were used, but mainly it is in 1910. That is the first antimicrobial drug, that is Selverson, not Selverson, that was used in the treatment of syphilis. And then in 1928, that is the Alexander Fleming, he discovered the penicillin. And then it is in 35, another, that is the sulfonamides. They were invented. They are antibacterials, not antibiotics. Antibacterials. They were invented. And in 1941, two colleagues, the Joshi and Prof, they conducted the clinical trials for the use of penicillin in the treatment of bacterial infections. And after that, it was widely used in 1943, that is World War II, and thousands of patients' lives were saved. In 1945, all three, the Alexander Fleming, Chain, and Prof, they were given Nobel Prize for that. In 1948, the resistance to penicillin was reported in Staphylococcus. 1953, they started using this antibiotics in animal feeds, and now they are trying to ban in animal feeds. So, understand the chronology. This is not only happening with antibiotics, but with everything. It can be the gases, refrigerant gases also, CFC, they have used, they have invented new, and they banned CFC. They played lot many with the atomic weapons, they will satisfy them, then banned it. Likewise, for the chemicals, for pesticides, they used plenty, they will satisfy, invented new ways, then they banned. So likewise, they started using antibiotics way back in 1953, and then the methicillin antibiotic, they were invented in 1959, and immediately they found that the methicillin resistance organisms, okay, this 59 and 16, immediately they got the methicillin resistance. In 1987, they invented daptomycin, that is the antibiotic, and after that, for 30 years, no antibiotic was invented unless till 2017. They invented new antibiotic in terms of the mode of action. Otherwise, every year 10-15 new antibiotics are added in the market. But you will find that they are the derivatives of the earlier antibiotics. Mode of action will remain all the same. So for 30 years, no antibiotics were invented. Then British started protesting against the use of antibiotics, and in 2017, the new antibiotic, that is the Teixobactin, that was invented. So, if you look at this, and then when Alexander Fleming was given the Nobel Prize, that time in the that receiving lecture itself, he told that very easily the antibiotic resistance can be developed. And simple way, he told that you give the low dose of penicillin to microbes and they will develop resistance to penicillin. If you see in brief that the discovery of antibiotic, and immediately preceding few years, you will find the resistance is developed. There is penicillin, tetracycline, and carbapenem, like that. Almost all antimicrobials, they are invented, and within next few years, you will find that resistant strains of bacteria, they are identified. What are the causes for AMR? So different factors are there: environmental, drug, physician, microbial genetics, patients, and how. In environment, you will find that rapid spread is an increased community acquired resistance and widespread of antibiotic in animal husbandry and agriculture. So that is the cause. Poor adherence to dose. Patients, many a time you will find that over-the-counter sale of antibiotics without prescription of physicians. Quality of drug, you will find that even though antibiotics are there, they are administering the patient or the patient only, be it human or animal. And you will find that whatever concentration written, same concentration you will not find in the oil. Okay, so low doses of drug or it may not be antibiotic at all. Okay, or very little of that one. So that is also cause inappropriate use of available drugs. So and physicians have a choice to give the antibiotic. Most time it happens that they prescribe the highest order of antibiotic, and that creates a problem. Once patient develop resistance against that, then difficult to cure the infections. And microbes themselves struggle very hard to survive, and they succeed by getting the genes from the neighborhood. They get the antibiotic resistance genes by conjugation, transfection, transformation, or number of ways they have, and they deploy all the means. So, as I told, every decade you will find new antibiotics are added. In one decade, put 2 antibiotics, and of late now their research has come down. So this has become a probe now. Antimicrobial resistance will be next pandemic, and new strategies. So the AMR is there, and how to combat that one. So new strategies are like this: plasmid curing, conjugation inhibitors, CRISPR-Cas technology, and Phages. So out of this, we will try to focus on Phages, how we can exploit Phages for the AMR. What Phage means? Phage is a virus which infects bacteria. Simple. Most abundant organism in biosphere, acellular, non-metabolic, and obligate parasite living organism. And for Phage, it is called that Phage follows culture. So wherever you have bacterial culture and against that Phage is there, you have not to make any attempts. Phage automatically find out where the culture is, where the food is. Bacteria is food for them. So they find out and they grow over there. So discovery is made by a British and a French, and both of them not only discovered, but after discovery they established the companies, commercial companies, and they sold varieties of Phage and Phage preparations. And they coined the term that this is bacteriophage, and it means bacteria eater. So, 1,000 types of phages which may infect only one or few type of archaea. So, it may happen, or it is likely that for a bacteria there can be varieties of phages may be there, but it's not there that for all bacteria there has to be a phage. There may not be phage. That is also possible. What is the life cycle of phage, or how they multiply? 2 pathways they follow: Lytic and Lysogenic cycles. When we say Lytic, it is the simplest one. The phage that will adhere on the host, then it will insert the nucleic material in the cell, that is bacteria. It will multiply. The nucleic acid will multiply. After multiplication, then they will synthesize the materials that is required for the construction of phage. Okay. So once that is synthesized, then the assembly is made, and once sufficient number of phages are there in the cell, then cell is ruptured, completely lysed, and all phages come out. Against that, the lysogenic cycle almost like this only modification, only changes. After the infection, you'll find that this nucleic acid, this material, may get integrated into the host genome. Okay, and it will get inserted into host genome, and before the actual lysis takes place, before there can be multiplication of the bacteria also. Okay, So bacteria will multiply, and in some of the bacterial host copies, you will find the lysis, and others with the integrated, as you know, they will be multiplying. After multiplication also, some will continue to multiply, some will continue to release the phages. Okay. So that is called lysogenic cycle. This is called Lytic cycle. What are pros and cons for that also we'll see. And when you say phage therapy, that is using the phage for the treatment of bacterial infections, phage therapy. So same thing, when the bacterial infections are there, you give the phage, and this phage will adsorb, and then DNA will enter into that, and there will be lysis of cell. That means the bacteria are killed by the phage. In the process, they release again number of phage particles, and each particle finds the new bacteria for adsorption, and again it continues like that. Okay. So that is the phage therapy, and this is the picture where if you have got a clone of bacterial cell, and then you apply the phage liquid containing phage on this one, diluted one, then you will find wherever phage is there you will find a transparent spots are developed. So at that place, the phage is multiplying and eating up or killing the bacteria. So that way you can get rid of the bacterial infection. Now, different ways you are identified. Phage. Shall we use, or is it mandatory to use whole phage? Yes, whole phage also can be used, or the components of phage can be used. Some genes the phage are having, so they also can be used, like the lysine, the protein that the phage are having, or the holins, and they are also the protein. The lysine that will cause the lysis the cell, whereas holins will create pores on the bacterial cell wall, and that way they will kill the cell. So in whole phage, one phage infects bacterium and release thousands of phages, and this phage can be used as a vector for delivery of lethal genes or antibiotic or bacteria. So in whole phage therapy also, you can use a phage as such, phage in combination with antibiotic, or these phages can be modified, engineered, genetically engineered, okay. And then also they can be used. Phage deficient in their lytic system replicate within and kill a target bacteria. So what is the limitation in this one? The phage, they are very specific, and they require bacteria. That means in Staphylococcus also, Staph aureus also, a particular strain is there. So the phage can be strain specific. And if they are acting on one strain, they may not act on other. So that specificity is phages are having. And that becomes the limitation, because if you are not in position to identify the actual causative agent that is infecting or that is causing the disease, cannot use. Okay, other ways are there. So whatever organism is causing infection, you isolate, and against that you identify or isolate the cause, and then use the cause for the treatment. There are successful isolation and treatment reports are there. That is because, but it's time taking. It will consume a lot of time, nearly 3 to 4 months. Time will go in isolating, identifying, and isolating the causes. So that becomes the limitation. Development of resistance to phage attachment. So what this means and how it happens, that also we'll see. And these are the phage components that I told you. Lysine that is having the bacteriolytic activity. So you can use this one for lysing the bacteria. Only thing is then it will behave like the drug. As much you are giving, that much only will be there, isn't it? When you are giving whole phage, it is replicating, multiplying of its own, and in that case, 1 or 2 doses may be sufficient. Okay, for one infection of one patient. Here you need to give till the infection is removed. Okay. Likewise for the pollens also for phage compounds. So these are the specific. This is the limitation for the phage and phage compounds. Now, if you compare the phage therapy versus antibiotic, then it's very, very clear. And also we'll see one by one. Phage therapy is highly specific, that I told that even for the Staphylococcus genus specific, you will not find phages that are specific for entire genus, but they will be even strain specific. So in that case, you need to have the wide repository of the phages, and whichever infection comes up, you need to prepare it. Antibiotics, they are having the broad range. Okay, so it can be against gram-positive, gram-negative, or both. I think both like that. Only pathogen can be targeted, so that is, you can consider advantage. When you are using phage, so it is targeting only pathogen, and the host, that is human or animal host, that is harboring the infection, is not affected. So that is the advantage. In case of antibiotics, it's not possible. So you are giving antibiotic, so it will act on the bacteria also, as well as it will have effect or side effects on the host also. It is bactericidal. Here it can be bactericidal as well as static. Bactericidal, that means we are considering the Lytic type of cycles. Effective on both antibiotic sensitive and resistance bacteria. So here it is only sensitive bacteria. Antibiotic will work for the bacteria that are sensitive to that antibiotic. Likewise, same you can apply over here. The phage sensitive only. You can have the phage therapy. Number increases till target bacteria are there, so they are self-replicating, because as long as the host is there, they will be multiplying. So single dose is sufficient. Here, antibiotics gets metabolized. So when you are injecting antibiotic in body, the body system, liver, it gets metabolized, degraded. Okay. So periodically you need to give or inject the antibiotics. Little increasing toxicity and side effect that creates it. So as such, phages are not having the toxicity or the side effect, whereas antibiotics do have. Minimal symbiosis of normal flora, because they are very specific and may disrupt the normal flora. So because they are in the broad range, So, be it the pathogen or the commensal, all are affected. Easy administration. Easy means administration means when the infections are superficial or bacteremia or like that is there in that condition. Problem with route of administration, it can be oral or intramuscular, IV like that. Safe, better tolerance and less expensive. Provided already it is available. Again, you have to newly isolate and produce it, then it is costly. Antibiotic depends on the penicillin, sulfonamides too, the higher range of antibiotic. So it can be cheap also or be costly. Only single or few doses are required, and here it is limited. No toxic, no toxicity to animal, plant, and the environment. So this is more important, and this is toxic to the environment also. Phage resistance is rare, is not. It's debatable. Okay, but antibiotic resistance is common. That is of sure. Takes few months, and development of antibiotic takes several years, and sometimes decades. So limitations of phage therapy: selection of appropriate phage. That is the limitations. Phage host range. Okay, for all bacteria you may not get the phages. What dose or amount of phage should be used every time you need to standardize. Right. Reduce activity due to immune system response. This is the challenge. Okay, when you are applying superficially, this may not be the challenge. But when you are administering for the infections that are deep-seated infections, in that case, when you are inserting phage, so body system will react to those phages also as a foreign elements, and against that also antibodies may be developed and they may be neutralized. May trigger immune system to overcome that, and there may not be enough kind of phages to treat all bacterial infections that we have seen. Some phages may cause bacteria to become resistant. We'll see this mechanism. Shifting of phages creates biosafety issues, and patenting issues discourages capitalists from investing. So this is the limitation, biggest limitation, that is you cannot patent phage. It's natural living organism, so it is out of range of patenting. So those who are investing in the industry, so they will not come forward for that for this purpose. So that is the biggest limitation for this one. Whereas in antibiotics, you can very well patent, and that's why many industries they are going for that. Now, this is the bottleneck. That is the CRISPR. What is CRISPR? All of you know now, okay, because these are all almost 20 years old things. CRISPR clustered regulatory interspersed short allotropic regions, and together with the CRISPR and CRISPR-associated gene Cas, it provides adaptive immunity against phages. So bacteria are having two types of immunity, like eukaryotes, human being, innate immunity, and the adaptive immunity. So which one is the innate immunity?
Skin barrier, saliva.
In bacteria
Restriction enzymes.
Bacteria, they are having also innate immunity. So restriction endonucleases, RE, in short. So any DNA that enters in the bacteria, they will chop using the REs. This is normally is not present. Okay, but as the infection, they encounter infection. Okay, cause infection. That time, some part of the phage genome, they will copy and insert in their genome. Okay, So this is the clustered regulatory interspace, short palindromic regions. So they copy that and put inside their genome. So it is memorized. And when next time that infection comes, so they copy it, they transcribe it, and then they form the CRISPR RNA, and then the Cas gene, put together this assembly. Whatever new reinfection occurs, okay, not the new reinfection. In the new infection, it copies this over here, okay. But when the reinfection occurs, they copy this one on the RNA, and that is guide RNA, and then they find the match in the Phage DNA or RNA, and then they bind over here and then cut it. Okay, so they neutralize the Phage infection. So if the CRISPR element is present, then and bacteria uses that system, then your Phage therapy becomes ineffective. Okay. So that is the limitation. CRISPR is having the natural mechanism of bacteria, but we are not remembering them. We are not knowing this. What we know about CRISPR-Cas, that is gene editing tool. Okay, so that is a tool that we have modified for other use, other purpose. But actually, it is the defense mechanism of the bacteria. Okay. So how to overcome Phage resistance? Different ways are there. The Phage cocktails you can use. So simple way, because these bacteria, they remember the previous exposure. Okay. So best thing is, don't allow any bacteria to survive. In the first go itself, you kill all bacteria. If they are not surviving, they are not going to remember. Okay. So no resistance will develop. Correct? For that, what you can do is use Phage cocktails. Okay. Or the Phage enzymes you can use. Only Phage enzymes you are using. No nucleic acid that you are treating with applying to the bacteria, so they will not develop the resistance against, isn't it? Because in CRISPR, what they are doing is they are taking the nucleic acid portion of the Phage and integrating in the genome. Here you are giving only the enzymes, Phage enzymes for the treatment. So no nucleic acid is there. So no fear of developing resistance is that. Okay. Phage and antibiotic synergy you use in combination. Okay. So give Phage also as well as antibiotic also, and you kill all the population of positive pathogens. Engineered phages, okay. So that also can be tried, and the Phage and the CRISPR, modified CRISPR, so that the bacteria is not copying the genome of the Phage in the system. Is it really happening like that? Okay. So FDA has approved a number of products, but all of them you will find they are for the food industry, either for the meat or poultry or like that food, or in agriculture. The treatments are there, fast treatments are there, they are available in the market, but they are not approved by the FDA. So that is the only limitation. These are few references from which the presentation was made. So, if you got any question, ask therapy. Right answer. Yes.
as you mentioned that FDA approved phage therapy is basically the oral administration route. So apart from oral administration,
Surface.
any other route of administration?
There's a surface cleaning, not oral.
Mostly so far it is for the exterior application, gel or like that. Some injectables are also there, like in mastitis also they are trying. Orally also can be given by injection also they can be given. But the problem is in all those applications the problem is the post immune system will be low and then it may develop antibody against that. That is. Otherwise, most applications for fast they are the exterior applications. Gels are there.
Topical.
Correct. Even for the food industry, it is the spraying out like that. Okay. So, exterior applications. Fine then. Thank you. Correct.